All right, so next we have Bhanu. Bhanu is a postdoc for the NASA program, and he prior did a postdoc with Dror Noy at Migal in Israel, working also on iron-sulfur protein design. And he's going to tell us about expressing iron-sulfur proteins in cells.
Hello everyone. My name is Bhanu Prakash and I'm going to talk about iron sulfur cluster proteins and peptides, artificial iron sulfur cluster proteins, and its implication in understanding the origins of life. So we aim to construct artificial electron transport chains from minimal protein building blocks. So here on the left, we see a natural protein, which is very large of more than 1,000 amino acids, and they have so many iron sulfur clusters and which can transfer electrons from one part of the enzyme to the other part. So, and in the middle, we see an artificial design protein, which is of about 100 amino acids and that has one iron sulfur cluster binding site and that can mimic an artificial and can mimic the natural enzyme. So basically, we are trying to minimize a really small protein from this big natural protein. Likewise, the theme one is also working on a prebiotic uh, peptides where we have very small peptides as small as 10 to 20 amino acids and have one iron sulfur cluster binding site. So for the artificial proteins, we employ computational protein design to have our de novo proteins. So for that, we basically take a three-dimensional structure from a PDB, protein data bank, and we predict a novel sequence. And this way we get many sequences. And this particular work is being carried out by Vic Nanda at Rutgers University and Rorna at Migal. Um, so why 4 iron and 4 sulfur cluster proteins? It's because of this relatively simplicity. It makes as an ideal templates for the de novo protein design. And also in the previous talk, Vic was highlighting about the importance of iron sulfur cluster peptides and its and as a role as a primordial catalyst. So if you look at the structure of the 4 iron 4 sulfur cluster, it's very simple. It has 4 irons and 4 sulfurs, and it is how it is arranged. And the 4 irons are bound to 4 cysteine atoms, 4 cysteine molecules. So if we this cysteine positions are highly conserved. So once we have this positions fixed and we have any random sequence in between, so we have a novel de novo protein. So for now, we have been working on helical systems. So on the left, we see a DJB system, um, which is a 4 helix bundle system. And in this case, there are 4 cysteines, and each cysteine is coming from 1 helix. So this has one 4 iron 4 sulfur cluster binding. So this is the side view, and this is the side view, and this is the top view. So likewise, we have another system called CCIS system, which is coil iron sulfur system. So in this system, which is again a 4 helical bundle system, but it has 2 systems coming from helix 1 and helix 3, and the other 2 helices like helix 2 and helix 4 are not having any systems. So it is not binding the cluster. So it overall makes a 4 helical system. So apart from the cysteine localization, there is another difference between these two systems. The DJB system, we can have the iron sulfur cluster assembled only with in vitro reconstitution, whereas the CCS system with this protein is we can have this assembly directly from the E. coli. Indeed, this is the first artificial protein that can bind an iron sulfur cluster in vivo inside an E. coli. And we have seen in the previous talk Saroj was talking about the DJB system, so I'm going to emphasize on CCIS system. So this is the CCIS system. As I told that this is the first artificial protein that can have an iron sulfur cluster directly from the E. coli. And this was the initial design, and these are the cysteine positions. On helix 1, 16, 13, and 17 are involved in iron sulfur cluster binding, and on helix 3, we have cysteine 61 and 65 position. This as per the initial design. And we get iron sulfur cluster directly assembled from the E. coli, and we have just to see the electronic structure, we have done the mass spectroscopy. For that, we have employed iron 57 isotope, and this is the textbook example of the iron sulfur cluster spectra, and the relative velocity for this spectra is about 0.44 millimeters per second, which is in the range of 4 iron 4 sulfur cluster proteins. And further on, we characterize its biological properties. This is the CD in the visible region, which is very typical to a 4 iron 4 sulfur cluster protein. And this is the UV visible spectra. This is little tricky because the 415 to 280 ratio is not very good as it should be. So there could be two possibilities. One is it could be a very low yield, or it could be a higher oligomeric state. So to confirm this, so we have performed size exclusion chromatography and also static light scattering. This is a static light scattering data. Here we see that the protein majority exists as a trimer. So and from the Ferrodoxin assay and from ICP-AES, we know that there is only one iron sulfur cluster in this trimer, not that there are three iron sulfur clusters. So the question is, what are the participating cysteines? To answer this question, so we have performed extensive mutational studies and found that only cysteine 13 and 17 of the helix 1 are involved in iron sulfur assembly, whereas cysteine 61 and 65 of helix 3 are not at all involved in iron sulfur cluster. So we tried crystallography. We couldn't get any crystals, but so we decided we'll do SARCs, which is small angle X-ray scattering. So this data was recorded at ESRF in Grenoble, and this is the envelope we got from the data. And so this is a triamic model that we built, and based on our mutational studies that we know that only helix 1 is binding the iron sulfur cluster, which is this part, which is the central part. So here we see all these three helix 1 of all the three monomers are converging at one place. So and this particular trimeric model fits almost very well in the envelope we got from the SARCs data. And this is a trimeric core. If we look from the top view, this is how it looks like. So the one of the helix is contributing two cysteines, whereas the other two helices are contributing one cysteine. So this is how the trimeric core looks like. And here we ask ourselves a question. So what happens if we truncate between helix 1 and helix 2? What happens if we incorporate a stop codon just after the helix 1? Will it assemble inside E. coli? Because this peptide is quite small. So for that, we have done the truncated mutation, and we got the truncated peptide, and we expressed in E. coli, and the answer is yes, it does bind an iron sulfur cluster inside an E. coli. So this is the smallest artificial peptide or a protein that we can make it assemble with an iron sulfur cluster directly from the E. coli. So likewise, we began to characterize as all what we have done for the CCS. For example, this is the UV visible spectra, which is very similar to the CCS. And this is the CD in the visible, which is again very similar to its parent CCIS. The CD in the far UV is slightly different, obviously, because it is 100% helix. And this is the Mossbar spectra. Again, this is also very close to its parent CCIS. So with this, we can say that the small helix is binding our iron sulfur cluster. So we thought that because there's a very small peptide and it's mostly highly structured because it's almost 100% helix, we thought we can try to crystallize it. So for that, we have been collaborating with Aki's group at University of California at San Diego. And Julian is a postdoc who is working on crystallization of this particular small peptides, which is single helix iron sulfur cluster. So here we got some small brown crystals. These crystals we got it like just few days back. So they're going to shoot them with X-rays and going to collect the data and hopefully solve the structure soon. So apart from the artificial proteins, the theme one is also trying to understand the ferrodoxin fold evolution. So here we are looking at ferrodoxin, which is a modern day bacterium ferrodoxin, which we can call it as like an asymmetric monomer. Basically, it has a monomer, but it has two iron sulfur clusters, and each domain is the sequence of each domain is very different. So we can call it as like an asymmetric monomer. So Paul and Vic, they've been working on understanding the ferrodoxin fold for like last few years. And if we see like how it has evolved, how it has originated as in a linear flow chart, this is how it looks like. So initially, it all started with a small peptide binding an iron sulfur, single 4-iron 4-sulfur cluster, which over a time, due to course of evolution, became a dimer, which we can call like a symmetric dimer. And over course of evolution, due to fusion, it became a symmetric ferrodoxin. So this is how it became a symmetric ferrodoxin. And again, over time, it further evolved to asymmetric ferrodoxin, in which each domain started evolving independently, and that's how we see as asymmetric ferrodoxins in modern day bacteria. So if we the first part, as we see the small peptide, this the ambidoxin has been already worked by John Kim and Pike, and was published in JACS. And recently, Andrew Murter, who was a former postdoc of Paul and Vic, he worked on the symmetric ferrodoxin design, and he published and they published in PNAS. So right now, I'm working on the peptide dimer, which is this part. So this should actually connect all the missing links between from how it all started with a single peptide with Iron-Sulfur cluster and today's modern asymmetric monomer ferrodoxin. So before I go with my talk, I just wanted to talk about symmetric ferrodoxin. The work was actually done by Andrew Mutter, and his work was recently published in PNAS. So he has 4 designs. So I took half of his designs. His designs for like almost up to 55 to 60 amino acids. So because I want to make a symmetric dimeric ferrodoxin, so I took half of the designs of Andrew's original designs and started working on it. So I'm going to show the data for the first 2 designs. So I had synthesized these peptides and started reconstituting with these peptides, and it was binding an alpha cluster to prove that whether it is a monomer or a dimer. So we have performed anaerobic FPLC, anaerobic size exclusion chromatography, and found that both of these peptides salutes as a dimer. And and looking at the ratio of 280 to 415, which is quite high, we can say that there is one cluster per monomer, and we see no monomeric peak, saying that the assembly is close to 100%. So now we have this assembled cluster. So to see the electronic structure, we have done EPR. For that, we have this data was recorded by Alexei, who is an EPR specialist at Rutgers University. So here we see both these peptides can be 100% reduced with sodium diethionate, and we have a very good spectrum of 4 and 4 sulfur cluster. And looking at the small bump here and here, we can say that there are 2 clusters and they are coupled to each other. So now we know that these peptides can bind to iron sulfur cluster and can transfer electrons in vitro. And now the question is whether these designs, do they transfer electrons inside an E. coli? For that, we have performed biological electron transfer assay. So for this assay, we have employed E. coli EW11 strain. So this strain is an auxotroph, and if we provide sulfite ion as a sole sulfur source, these bacteria cannot survive. So for this bacteria to survive, we should introduce a ferrodoxin gene, and that gene should be successfully able to transfer the electrons from the FNR to the ferrodoxin protein, and the ferrodoxin is transferring the electrons to the SIR, which is sulfite reductase, which reduces the sulfite ion to sulfide ion, which is S-2. And the E. coli EW11 strain can employ this sulfide ion to make amino acids like cysteine and methionine and other important biomolecules. So as I'm saying that this ferrodoxin gene is transferred directly into the bacteria through transformation, and this is inducible with ATC, which is anhydrous tetracycline. So this work we're doing in collaboration with Silber's group, and Ian Campbell is a PhD student who is working on this project, and he got some exciting results for us. So here is the results from Ian Campbell. So the top panel, we are looking at a lot of controls. The first 3 being the positive controls, and the last one, this one is a negative control. So what happens when we have ML ferrodoxin, the wild type ferrodoxins, when we have, you see that in presence of ATC, the E. coli cells can be rescued very fast. And this is SCC, which is Andrew's design, one of the which is symmetric monomeric ferrodoxin design that can also transfer electrons in vivo and can rescue these E. coli cells. And this is an uninoculated cell, which is basically a negative control. Here there is no ferrodoxin gene at all. So even in presence of ATC, there is no rescue of the E. coli cells, and the cells will eventually die. And the bottom panel, all the 4 are new ferrodoxin have designs. So in all these cases, we see in presence of ATC, there all of this, in all cases, the cells are being rescued, meaning that these all 4 designs can transfer electrons even inside an E. coli. So with these results, we can say that the ferrodoxin indeed originally originated from being a small peptide binding a single iron sulfur cluster, which later on became a dimer, and over course of fusion it became a symmetric monomer, and again over a course of evolution due to independent evolution of each of the domain, it became a symmetric monomer as we see in modern day bacteria. So apart from the artificial protein design and ferrodoxin fold, the theme one is also working on prebiotic chemistry, where, for example, I'm talking about Ambidox, and we had seen already Vic talking about it. So I'm going to talk about it very briefly. So this is a very successful design from Vic and Paul's group. So here we have amdoxin, which is 12 amino acids only, which is a very small peptide having L and D amino acid and has 4 cysteines and can bind 1 iron sulfur cluster. So this is a computational design. And as per the design, it is binding an iron sulfur cluster, has 1 iron sulfur cluster binding site as evident from EPR data, and it can transfer electrons, and it can perform robust redox. It can undergo robust oxidation and reduction cycles over thousands of cycles. So this work was done by Dog Pike and John Kim and was published in JACS. So Dog, who initially designed this ambidoxin, later further began to design uh, more designs. For example, like more simplistic designs like a mono iron binding ambidoxin, and also like a 2 iron 2 sulfur cluster binding ambidoxin, and also complex clusters like the P cluster as we see in nitrogenase. So these are all computational designs already, and Jenny is a new postdoc who joined our group, and she's going to work on these designs, and she even synthesized some of these designs, and she will be characterizing these peptides very soon. And thanks to all my professors, collaborators, and friends in USA, Germany, and Israel. Thank you. Thanks for your time.